Hello, everyone. My name is Ivan Pires, and I'm CSO and co-founder for the Hammer Nervite team, where uh, we're revolutionizing immunotherapy of perennially disseminated diseases, taking what was previously incurable diagnoses and transforming them into routine care. Oh. Our need is we're trying to find an experienced CEO as well as individuals with regulatory experience. Moreover, we're trying to establish strategic alliances with anti-PD-1 checkpoint inhibitor companies, and we're initiating our seed Series A round of $50 million to propel us to the next stages of development. So the main indication that our technology is initially targeting is ovarian cancer, which is a clear example of failure of therapeutics development. A woman's lifetime risk of not only being diagnosed, but actually succumbing to ovarian cancer is of 1 in 100. And in the US alone, over 20,000 women will be diagnosed with ovarian cancer, and this often comes at a late stage, meaning that their survival rate is of less than 30% in 5 years. And while there's been many successes for development of therapeutics in many cancers, ovarian cancer has completely failed, such that patients today currently rely on just surgery and chemotherapy for their treatment. We're trying to change that by introducing our first product called OncoLatch, which can target disseminated peritoneal metastases, delivering a potent immunostimulant such that we can kickstart the immune response and have immunotherapy be a reality for ovarian cancer patients today. Just a little bit more about our OncoLatch. We're pioneering a new targeted, non-internalizing nanoparticle platform that can target these peritoneally disseminated metastases. And how it works is we deliver a cytokine called interleukin-12, which has been validated in the numerous preclinical studies to be the cytokine that you want to be in the tumor. What we do with this cytokine is we conjugate it to our nanoparticle, and then we coat this nanoparticle with a proprietary polymer coating that allows us to have the two key properties that are the targeting and the non-internalizing. These particles are injected intraperitoneally into these patients that have this metastatic disease, where the particles can go, surge, find, and bind to these tumor cells while sparing your healthy tissues. And really, the turnkey solution for the delivery of interleukin-12 is this non-internalizing phenotype, whereas OncoLatch is able to bind to these cancer cells and stick to their outside, where they can signal to the immune system for prolonged periods of time, such that you can have a strong immune response that kills off the tumor. Alternative strategies, such as your standard antibodies targeting system or standard nanoparticles, once they bind to the cancer cell, they're internalized, and their cargo is taken into the cell with it, such that the immune system can no longer be activated by the cargo. In preclinical models of the highly metastatic ovarian cancer model, where animals end up succumbing to the disease within 20 days, we can try to treat this animal with your current standard of care, such as chemotherapy, or even we can go beyond and try to use the best immunotherapy combination, which is your anti-PD-1 and anti-CTLA-4, and you can see that this treatments completely fail in this model. We can try to now combine the free cytokine with checkpoint inhibitors, where we start to now see some responders. If we now just use OncoLatch to deliver interleukin-12 on its own, we can already start outperforming this combination therapy. But what was really exciting is the moment we use OncoLatch and combine it with checkpoint inhibitors, where we can completely cure all the animals in this model from this highly metastatic, chemo-resistant ovarian cancer model. So we can achieve this over 88% survival in this ovarian cancer model. We've been able to see similar results in additional ovarian cancer models. We are not able to detect any long-term side effects. We actually have the abscopal effect that you heard before in the first presentation from today. And we actually vaccinate these animals from recurrence by producing long-term immunity in these animals. And what's very exciting about our future is that our targeting strategy is a pan-cancer targeting, meaning we can rapidly expand to new indications with the same product. And while we're initially starting with these chemo-resistant late-stage ovarian cancer patients, which on its own has a market of over $2.5 billion, we'll expand this initially to just making this product a first-line adjuvant for ovarian cancer patients, but then we can rapidly expand to other peritoneally disseminated cancers, such as colon, pancreatic, and gastric, just to mention a few. And then more broadly, we can actually take this and use it as a therapeutic platform for many other cargoes, as while I'm initially focusing on OncoLatch, which has this surface binding property, we've already identified polymer chemistries that allows us to target cancer cells and actually deliver cargoes intracellularly, such as, for example, your standard chemo-based therapies. To make this product a reality, we've assembled a team of highly experienced experts in the field, both in the preclinical space and in the clinical. The advisors On the preclinical side, it's composed of myself and my advisors from during the PhD at MIT, where I worked on this product, and they're composed of Daryl Irvin and Paula Hammond, who bring a lot of the key nanoparticle development and immuno-oncology experience. And on the clinical side, we have Dr. Kevin Elias and Dr. Ursula Matulonis, who both have been leading many of the key clinical trials in the ovarian cancer space to the development of new therapies for these patients. And in the next couple of years, we're going to have many key exciting milestones to be reached. We're going to be continuing to develop this product in the preclinical space to expand our IP base and demonstrate its effectiveness in new indications. We've already secured a $1 million DoD grant. We're awaiting likely a positive result, or at least I'm hoping, from that NIH. And we're also planning on applying for SBIR and STTRs. And the next year, we're going to have our initial tech transfer and our first dose-ranging NHP study. We'll then progress to make these protocols under a GLP so that we can have our initial IND tox study. And then we'll actually come in, produce this under GMP so that we can have our IND filing, and then reach our first human proof of concept with this product by the end of 2029, early 2030s. And we've already been able to identify the key partners who are going to be working with us to make this a reality. And that is Cytiva, who's going to be helping us with the nanoparticle, Helix, who's going to be working with our interleukin-12, and Charles River, who's going to be the one running our animal experiments. So coming on back to our asks, we're trying to find an experienced CEO who has taken these products like this from the early stage through IND and into clinical studies. We're also trying to find individuals who have worked in the regulatory space to help us with the filings. Within our strategic alliances, we're trying to find suppliers of anti-PD-1 who can help us both in the preclinical and clinical development, as well as companies that are developing their checkpoint inhibitors who want to co-develop with us to expand their indications and their treatment modalities. So thank you for your time. I hope that what you can take away from this is just with $15 million, we can start to revolutionize ovarian cancer care. I hope that I can see you in my table later today. It's table 11, and I look forward to talking to you all.